Have you ever wanted to control something with your mind? For example, to change the channels on TV, or maybe to dim the lights a little bit, or to send a text message just by thinking about it. So the way to potentially do this is by using brain-computer interface, or in short, BCIs. And these are systems that can read the brain activity and then translate it into a machine or a computer language and send an external feedback to the one who uses this. Okay, so right now we're not actually using this system just to, for example, think about the coffee machine to activate whenever we want some coffee. But I will show you my attempts to do something that is quite similar to that. Okay, so nowadays brain-computer interfaces are mainly known for their clinical purposes. For example, this disabled woman is using her brain activity in order to activate a robotic arm that fetch her the drink she wants. But BCIs are not only used for clinical purposes, they are also used for research purposes. And we do that by using lab animals, for example, monkeys or mice, in order to better understand the learning processes that happen in our brain. Okay, so what does it take for a BCI? So each BCI has three main components. The first one is the hardware component that collects the data from the brain. The second one is a software component that translates and processes this data to a language that a computer or a machine can understand. And the third one is the generation of an action in the outside world. Okay, so nowadays, usually BCIs use electrical signals that are generated by neurons, by brain cells that are called neurons. So in this example, you can see there's an electrode inserted into the brain, and it collects the electrical signals from the neurons. So what we get is this list of electrical signals from all the neurons that were recorded. But in this method, you cannot track the same cells over days. And why is that important? So to remind you, a major part in BCI is the encoding part. So if the neurons keep changing between days, then the signals changing between days. So we actually need to teach our software over and over what are the signals and what it needs to do in order to generate the action. And not only that, for many research purposes, we are very much interested in what exactly the cells are doing over time. So we can ask questions about the dynamics of learning happening in the brain. Okay, so one approach to solve this is not using the electrical signal, but rather to image the cells themselves. And when we image the cells, we actually see them, and then we can track their identity over days. So the example I'm showing you here is of a miniature microscope that was designed to record the activity from mice brains. And this is the tool we're using at the Ziv Lab. And you can see here an example from 1,200 neurons that were recorded using this microscope. Each dot here is a different neuron. So with this approach, we can account for a large group of neurons, but also to track the same neurons over days and even months. So what's the problem with this now? In order to detect the single neuron's activity, we actually need to use image processing. So let me show you an example. This is the raw data coming from this microscope. And I want to show you what it looks like. Okay, so you can see that it's dynamic because there's an activity. This is a brain tissue from a mouse performing some task. And you can see here some dots glowing and turning off. These are the neurons that are active. But it is not trivial to extract this single cell activity on real time. Okay, so what I did during my PhD work was to develop this BCI that works with exactly this kind of data. So these mice, as you can see here, are really small, and they can allow the mice to freely behave on a variety of tasks. And the system I developed can read the data from the microscope on real time, process the image and the specific cell activity, and then trigger a reward system on real time according to specific neuronal patterns. And not only that, but because we can track the cell's identity over days, we can use the same set of cells to activate this BCI. Okay, so I will show you a short demonstration of an experiment we run in which the mice were trained to run back and forth on this linear track to collect water rewards at its ends. So you see here on the bottom panel, the mouse will run back and forth to collect the water rewards. But what I did was to choose this subset of cells that will activate the BCI. And you can see them in red here. But once they will turn green, the BCI detected that they were active. And then, in order to receive a reward, the mouse will need to activate at least 2 of these cells, no matter which. And you will see a green point here. Okay, so let's see the movie so you can see if the mouse managed to get his coffee. Sorry, water. Okay, so there's a green dot here. He managed to receive its reward. And then he received it again. Okay, so the mouse runs back and forth, and let me show you what happened across days. So in this graph, you can see a time course of an entire recording session, where the line here is the running trajectory of the mouse, and the red stars are where it received its reward. So you can see that in approximately 60% of the edge visits, it received its rewards. But not only that, when we look at the amount of reward in 6 recording session days, and to remind you, we are using the same set of cells across all of these days, the mice, three mice managed to maintain a rather stable amount of rewards across these days. But not only that, now we have this tool in which we can track the same set of cells, and we can better understand what are the dynamics of this learning across time, not only in our chosen cells, but also in the rest of the population. Okay, so to summarize, BCIs are used both for clinical and research purposes. And current techniques don't allow the use of the same cells over many days. So I developed a calcium imaging technique, BCI, that enables to do so, to do it with the same cells over many days. And we have our preliminary results that this system works and that mice actually learn to self-administer water rewards across many days. So it's not yet coffee, but for the mice, water is a pretty good reward. And with that, I would like to thank the Yaniv Ziv Lab. I would like to thank my mentor, and Alon, Or, and Nitzan, who are major persons in this, major contributors to this project.